Hello and welcome to the 13th lecture, MD Simulation Structure File. So we have started the discussion on MD software and servers, and we'll show you how to set up and visualize MD results. So previously we have introduced you to the MD software. Now we will particularly introduce you to the concept of MD Simulation Structure File that you will deal with. And we have already told you that in an MD simulation, when you run through a software, there are three types of things that you have to take care about. One is the interface that you are interacting with of a software that might have a GUI, graphical user interface, or a command line interface. And inside that package, there are packages of force fields that you will apply on your system. And at the end, you will visualize your result. So to begin with, you start with a structure file, the structure file of the material or the biomolecular system that you want to simulate. What is in that structure file? There are actually coordinates of the atom. And if it's a crystal, there are probably additional information like crystallographic information. If it's a protein or a biomolecule, then probably there will be more information, like which portion are helix, which portion are beta sheet, something like that. So a structure file actually defines the structure that you are going to use for your simulation. And in the visualization, what you are looking at, you see some balls like atoms, or you may see the secondary structure of a protein like alpha helix or beta sheet. In the visualization software, you'll see how does it look like, how does the molecular dynamics simulation and the updates of the trajectories when the molecular dynamics simulation proceeds, how does it look like? That information is you are getting from the visualization software. So when it comes to a structure file, the ending of the or the naming of these structural files differs based on what are they related to. For example, in material science, the most used files are with these endings, like these are probably XYZ file or CIF file, crystallographic information file. And if it is biomolecules, proteins, or membranes, you're probably going to start with a PDB file, a protein data bank file that contains the information's coordinates and the secondary structure information of the structure. Sometimes it's a platform specific file. For example, if you are working in LAMMPS package or the LAMMPS platform, you have to convert whatever structure you have to a LAMMPS data file. It doesn't have a definite ending. You have to convert it to a LAMMPS data file, and it has its own syntax and format, and you have to convert it according to that. Now, how do I open or read or visualize these files? There are several options for that. Those are also particular to your system or your approach to solve the problems according to your field. Several popular options are VMD, Chimera, Ovido, Vista. For example, a biomolecular system, generally you use VMD or Chimera. For material science, generally use Ovido or Vista. So long story short, in material science, generally you start with a structure file with XYZ or CIF. And if it's a biomolecule, you start with PDB. If it's a platform specific, you have to convert it to a LAMMPS data file. So how does it look like? Let's look at some examples. For example, I have taken it from the Wikipedia page of XYZ file. If you search for XYZ format, how does it look like? You can see that for the pyridine molecule, the XYZ file, if you open it with a text editor, it will look like this. It will show you how many atoms are there. You can see that there are 11 carbon, nitrogen, and hydrogen atoms, and you will see their coordinates in the 3D space, the X, Y, Z, and coordinates of the 11 atoms. So that's how an XYZ format looks like. It's really simple to interpret. If it comes to a PDB file, let's say for a protein, it's actually an extract from a protein PDB file. You can see that it will show so many information. For example, it will show the indexes of the atom. You can see that there are 12 atoms here. It will show there are nitrogen, carbon, oxygen, and what kind of carbons are there. and what kind of nitrogen are there, and what residue it belongs to, for example, lysine, for example, valine, and which chain it belongs to, and which residue it belongs to. You can see that the 11111, that is actually the lysine residue, and here the valine is beginning, and the actual PDB file is actually much more, which cannot be accommodated in this single slide. And you can see there are coordinates as well, which atoms are there, as well as there are other additional information. So when you open a PDB file with a text editor, that's what you look at. You can get so many information about this structure that you are going to simulate. And if it comes to a LAMMPS data file, it also has similar type of information. For example, it will start with how many atoms, bonds, and angles are there, how many types of these are there, and what is the box size in terms of X, Y, and Z coordinates. It will show you the box size in terms of angstrom or whatever unit you have used. What masses are there? You can see that there are 16 and 1, that is oxygen and hydrogen are there. There will be some molecular dynamics pair coefficients and bond coefficients and angle coefficients are there. Then there will be atom information. For example, there are indexes, there are coordinates, and so many more information. So that's how actually a LAMMPS data file looks like. This is actually a structure file that gives you an information of all the atoms contained in that system, in the structure. So where do you get this structure file? Sometimes you have the ability to create it from the scratch inside the platform. For example, in LAMMPS, you can create the in LAMMPS or VMD, whether you are working on a material science project or a biomolecular project, you can start it from the scratch. Sometimes it's not possible. Sometimes you have to go through some database. For example, popular material science databases are material science project, open material science database, crystallographic database, or specific research groups that make their particularly useful structure files and deposit in their repositories that can be accessed publicly or sometimes upon request. And if it comes to biomolecules, RCSD is possibly one of the biggest databases for structure files, biomolecular structure files. Sometimes you get the structure files from a UniProt or PubChem, and again, from specific research groups, repositories. So the structure file sources can be diverse and multifarious, and you have to determine how you want to make your system or how you want to start with your structure. So these are some homepage screenshots for these databases that I have told you about. For example, this is a material science project, which is very popular for material science related projects. You can get a lot of structures from there. Probably you have to sign in or register to download the structures in XYZ or CIF format. This is open materials database. You have to search for a particular alloy or an element, and when you submit your search, you will see how many structures associated with your search are available in their database, something like that. This is crystallographic open database where a lot of crystallographic information and structures are deposited. And this is a very popular one, RCSD, where protein and protein-protein interaction structures are deposited. For example, you can see here is a structure of hen egg white lysozyme that is a protein, and you can download this file in a PDB format or FASTA sequence, the amino acid sequence from here. And also this is UniProt. Here you can search for it. For example, this is also lysozyme. Chick means it's for hen lysozyme that I have shown you here. And you can also download in a format from here. And also there are a lot of additional information when you search for a particular protein or molecule here, as well as any other database. Feel free to explore through them, because a lot of information can be achieved from there. For example, how this structure is obtained from X-ray diffraction, and there will be the publication associated with it so that you can learn from it. And this is a very popular chemistry related database, that is PubChem. You can get a lot of chemical structure from here, aspirin. Sometimes you have to download the structure and convert it to another format. For this kind of conversions, you also have other software or facilities like OpenBabel or so on. And you can see that there are so many other functionalities and tons of information associated with a particular molecule. And this is an example of a structural file source from a specific research group. For example, this is JBKL lab of molecular and thermodynamics modeling that actually has deposited so many lipid membrane coordinates that they have worked on, and you can download it from there in different formats like PDB, CRD, PSF. So that's how we can get your structure files or make your structure files. And in the next lecture, we will talk about visualization of the simulation.